Thank you all for coming. Welcome once again to the Whom You Love Symposium Series. I'm glad to see a pretty good turnout, given that we had to cancel last week. I didn't tell you a lot about, I tried to email all the avenues I knew, and of course, so there's several ways you can keep track of the series. One is if you're a Facebook person, you can like Whom You Love, and that way you'll learn about things, for example, when we had to cancel the lecture last week. And the other is to go to the website, whomyoulove.com, and we'll post everything there. So my apologies if there were some folks that came to the lecture last week who didn't get the word in time that we had to cancel. I'm sorry to say that Dr. Bailey was hit by a car on the Friday before his talk was scheduled, and that he's okay, but he needed some surgery on Monday, and he's still got some recovery to do. We do still hope to bring him here for this talk. We'll see how his recovery goes. He's anxious to be here. He's part of this big project, the genetic screen for sexual orientation that's going on in Chicago now, and I'm hearing rumors that they're going to have some interesting things to say, so I'm hoping he'll get here to give you a preview of that information. So again, to keep track, and I sent out word to the remaining speakers that they would be especially careful around cars until after they've given their talk. And after that, they can be as reckless as they like. So whomyoulove.com or Facebook Whom You Love, and that way you can keep track. We do have to cancel things like that. So I'm especially happy to introduce Ray Blanchard today, who is, in my opinion, one of the giants in terms of research into sexual orientation and sexual behavior in humans. He got his degree actually studying learning in pigeons at the University of Illinois, and then eventually went into the clinical arm of psychology. And I don't know if he'll be talking about this too much in his talk, but his first job was working with inmates in a prison, which included sexual offenders, and he didn't really intend to become a scientist, but got exposed to the right guy at the right time to show him the sorts of things that could be done, sorts of things, sort of questions that could be answered, and I think we're all the luckier for it. He is also known for having discovered the older brother effect that he's going to talk to you about today. Is a very, to my mind, I still think it's a very mysterious effect, and yet it's so firmly established, there's absolutely no doubt that it's there. And it's, I would say it's one of the most important facts that nobody seems to know about sexual orientation. I think I would count it as that. Is that it's so firmly established in the literature, has been for over 10 years now, and yet when you talk to people in the street, they don't know about it. And so I'm delighted he's going to be able to talk to you about it. So please help me welcome Dr. Ray Blanchard.
I'd like to thank you, Mark, for organizing this lecture series and for asking me to be part of it. So I'm happy to be here. I've never been to Michigan State before, and I was happy to have this opportunity to see the campus. To put this talk in context, there are three broad classes of biological theories of homosexuality. The first one is based on the supposition that inherited genes influence a person's probability of developing a homosexual orientation. And evidence for this view would include the greater similarity of sexual orientation in identical twins compared with fraternal twins. Secondly, the tendency for homosexuality to run in certain families, and more recently, molecular DNA data. And the genetic theory has prompted various auxiliary hypotheses to explain why a genetic variant that's associated with such a low fertility level has not been flushed out of the population. And these auxiliary hypotheses include the kin selection hypothesis and the sexually antagonistic gene hypothesis, which Paul Vasey spoke about here earlier in this lecture series. The second broad class of theories is based on the supposition that a fetus's sexual orientation is influenced by its level of exposure or its level of sensitivity to prenatal sex hormones. This general area of theorizing lies within Dr. Breedlove's specialty, behavioral neuroendocrinology. And this hormonal supposition is based in part on the huge amount of animal experimentation in which prenatal hormones or perinatal hormones are manipulated directly, and by research on human clinical populations in which the fetus was known to have experienced grossly atypical levels of fetal hormones during fetal development. The third type of biological theory is the newest and the least well known, and that is the notion that sexual orientation in men, but not in women, is influenced by the mother's immune responses toward their fetuses. A version of this theory was advanced as early as 1981, but most of the research that's been based on it has been done by myself and my colleagues in Toronto over the past 20 years. So these data and the theory that I've advanced to explain it are the topic of the lecture this afternoon. When I prepared this lecture for today, I decided to, I made a couple decisions about how I would organize it. One is that I'm going to leave out some of the relevant research findings in hopes that what I've left in the talk can be presented more clearly. I also decided to organize the material to make the strongest possible distinction between the observed facts and the theories that have been advanced to explain the facts. And I'm going to start with the facts, as my PowerPoint slide subtly suggests. My third decision was that I would present the take-home message of each section at the beginning of the section rather than at the end, so that you can get the take-home message, and then if you space out or if I'm incoherent during the actual exposition of that section, you'll still have gotten the main point. This is the first and perhaps the most important take-home message, which is that older brothers increase the odds of homosexuality in later-born males. However, the other classes of siblings, older sisters, younger brothers, and younger sisters, have no effect on the probability of homosexuality in later-born males. I do not intend today to go over the minutiae of individual studies, which would be more painful for you even than it would be for me. But I've put some of these kinds of details up on the screen more or less as wallpaper to have up there while I'm talking about what some of the main findings are. This particular study, the 96 study with Tony Bogart, was a key study in that it was the first one to show that the tendency for gay men to be born late in their sibships was primarily related to their number of older brothers. In this study, I've got two screens here, this one and the next one, which turns out to be less helpful to me than I thought. We dichotomized men's sexual orientation as heterosexual or homosexual, and then we attempted to predict sexual orientation from the 6 variables I have up here, the 4 classes of siblings and parental age at the time of the mother's birth. The only variable that predicted a man's sexual orientation was his number of older brothers. I've highlighted the P level there for no particular reason, but it just shows you the number of older brothers was the only predictor, with everything else controlled for. And this slide shows that the odds, each older brother increased the odds of homosexuality by 33%. Now, I have to jump in here and say that to a statistician, odds and probabilities do not mean the same thing. I'm not going to try and explain to you the difference between odds and probabilities, but I mention the odds here because they're going to come up in a later slide. However, when I'm going to give you information that you need to understand at an intuitive level, I will talk in terms of probabilities rather than in terms of odds. This slide, this slide in which I highlight a different section of the data, was to, I think, emphasize one of the more important of the negative findings, which is that when you've controlled for siblings, then father's age at the time of the subject's birth and the mother's age at the time of the subject's birth are no longer significantly related to sexual orientation. This is an important negative finding because there are a lot of other traits, mostly negative traits, clinical conditions, which do correlate with advanced maternal age or advanced paternal age. But that does not appear to be the case here once you've taken SIBship parameters into consideration. It's just older brothers. This is another view of the data from the same study. Now, in the slide I showed earlier, it showed, but I didn't emphasize it, exactly half of the subjects, 302, were homosexual, and the other half were 302 heterosexuals. They were individually matched. Now, because the number of heterosexual and homosexual subjects was exactly equal, if there were no relation between homosexuality and older brothers, you would see a flat line that runs, a more or less flat line that would run through the 50% point here. That's what we'd have. Instead, what we see is a 45% probability of homosexuality, roughly 45%, for the subjects with no older brothers, rising to roughly 70-something% for the subjects with 4 or more older brothers. Of course, in a real-life sample, if I did a random sample and just let, without trying to have an equal number of heterosexual and homosexual cases, the data would not look quite like this, because in the population as a whole, you don't have 50% gay men and 50% straight men. So you can ask, well, what would this curve look like if we did a random sample from the general population? We can estimate what the data are going to look like using one of a system of equations that were developed by a guy named James Cantor, who at that time was one of my postdocs. And the equation uses the odds ratio that I showed you previously. That's why I mentioned, emphasized that there is such a thing as an odds ratio, because it's part of this equation here. So the one part of the equation comes from the study, the 96 study with Tony Bogart, the statistic that each older brother increases the odds by 33%. I think it's a decent estimate, because later studies sometimes produced odds ratios a little higher and sometimes produced odds ratios a little lower. So I'm comfortable saying let's proceed as if the true odds ratio is an increase of 33% with each older brother. The other thing we need for this equation is to say, what is the baseline prevalence of homosexuality in men with 0 older brothers? Of course, nobody has collected such data, really. Nobody goes out and says, I'm going to recruit guys with no older brothers, and let's see what proportion of them are gay. So as a guesstimate, I picked 2%, because it's a nice round number, and because it's in the window of what the overall population prevalence of homosexuality is in men. So I'm plugging the figure of 2% base rate homosexuality for men with no older brothers in here, and the odds ratio of 33% increase per older brother there. Having done that, we get this curve. And what the curve, I should say up front that I would never have generated this curve left alone, because it just would have seemed to me like a strange thing to do. But I kept getting asked over and over again by people who wanted this information. So I finally actually calculated these data, and then I started using them in talks like this one. What this shows is that if your baseline probability of homosexuality for men with 0 older brothers is 2%, it rises to almost exactly 6% for men with 4 older brothers. And I didn't realize this when I first made the graph, but it's almost exactly 10% for men with 6 older brothers. Now, I don't really believe that this is necessarily what the real life curve would be once you get out to men with more than 5 or 6 older brothers. I'm pretty sure that this is a reasonable approximation of what the curve looks like within the range of families that you would get in real life if you take a big sample. The rest of the curve, this part here, is an extrapolation that's what the curve would look like if what's true here continues in the same way. But it might not really look like that, because there could be some kind of ceiling effect or saturation effect whereby really the curve looks like here and then starts to not go up anymore after a certain point. I don't know. But people always wanted to know, well, how many older brothers would you have to have before you got to 50%? And the answer is more than a woman could have. Naturally, people always ask, what is the relevance of this phenomenon to homosexuality in women? And the answer is none. No class of siblings has consistently been shown to influence the odds of homosexuality in females. There have been several studies that tried to look at this, and the findings have been all over the map, which I interpret as a signal from nature that there is nothing. When I see findings that are this way and that way, and sometimes it's older sisters, and sometimes it's older brothers, and sometimes it's younger or this, and it's significant in that way or significant, then I know there's nothing. It's all sampling error. So I'm comfortably prepared to say there is nothing, that the fraternal birth order effect is a phenomenon that is caused by males, affects males, and females are just not involved in this particular process. And that conclusion is actually consistent with the theory that I will present later about why older brothers affect sexual orientation in later born males. Here, I just, again, this is sort of wallpaper. This is an exemplary study of showing no results for females. The original data were collected by a professor at Minot State University, and he collected these data long before I got involved with his project. So he basically had finished his study and collected his data, and then I asked him, or he asked me, I don't remember, if I would analyze the birth order data, which I did. He's got this humongous number of heterosexual women because he recruited his heterosexuals from university psychology and sociology departments, and they're quite often heavily skewed in terms of enrollment by females. Now, don't even try to understand this graph, because I just want to show you that there's see the big blue thing? Okay, the big blue thing is older brothers for gay men. All the other bars that are not as outstanding as the big blue thing are older sisters, younger brothers, and younger sisters for heterosexual and homosexual men and women. So we have all of the possible different kinds of groups here with all of the different kinds of siblings, and the only thing that sticks out from the crowd of bar graphs is older brothers for gay men. Okay. Now, the next take-home message, which is important and will be important for theory, is that it's only biological older brothers that increase the odds of homosexuality in later-born males. In contrast, stepbrothers or adoptive brothers, in other words, other boys that were reared in the same home as you who would have the social relation of an older brother, they don't do anything at all. It's biological older brothers that matter. There's only been one study on this, which was conducted by Tony Bogart, who is now a full-fledged professor in his own right. And he had to recruit different kinds of families, blended families. So he was recruiting people from intact nuclear families, from families that had stepbrothers or adopted children, so that he could tease out the effect of the difference between the effects of biological older brothers versus social older brothers. So it's an arduous kind of research, because you've got to be recruiting all of these different kinds of families from gay and straight men. Fortunately, the results were quite clear-cut and showed that it was only biological brothers that influenced the odds of homosexuality in later-born males. This, I don't want to get into theory just yet, but I think I should jump ahead and a step and say the implication of this is that older brothers influence younger brothers while the younger brothers are still in utero. And this result argues against the theories that have been explained that, for example, the reason older brothers influence sexual orientation is because there's sex play between brothers, and this somehow causes conditioning in the younger brother because he's younger and more impressionable. And there are other kinds of psychological theories that have been advanced to try and have an explanation of the fraternal birth order effect in terms of environment of rearing. These are more or less desperate turf battles to keep things in the behavioral realm and out of the biological realm. In terms of whether or not they're flattering to gay people, they're actually worse than the hypothesis I'm going to present. One of them that always stuck in my mind was somehow that younger brothers compare themselves so unfavorably to their older brothers that they become gay, which did not, make a lot of sense to me, but you can find anything in print. This is a finding that I put up partly, is this an homage to Mark because I used one of his data sets in doing this. I analyzed a lot of data sets, a lot of data where there was information in the data set on sexual orientation and handedness and sibship composition. And to my astonishment, and what I was expecting was that handedness would have an additive effect to older brothers, because we know there's some effect of handedness or some correlation between handedness and homosexuality. In fact, I found the exact opposite of the thing I was looking for, which seems to be that it looks like maybe older brothers only affect sexual orientation in right-handed males. Here is kind of a idealized representation of what the data look like, not a resume of data from one specific study. This green curve here is just like the curve I showed you earlier, relating the probability of homosexuality to number of older brothers. So you've seen this curve in an earlier slide. And this is what the data look like, as best I can tell, for left-handed guys. Their probability of homosexuality stays the same regardless of how many older brothers they have. I deliberately drew these lines to cross for reasons that I've just decided not to try to explain. But I think, but it has to do with the fact that there are other studies suggest that there has to be some crossover point at which left-handers become more likely to be gay than right-handers, and it would be at the point of 0 or few older brothers. Now, people always want to know about effect sizes, and the way I decided to approach that here is to pose the question, of all the gay men who exist, how many are gay because of the fraternal birth order effect? We did 2 studies on this, and the results suggest that the number of gay men who are gay because of older brothers, it was estimated as about 15% in one study and about 30% in another study. This is just a graphical representation of the 15 and 30%, which I basically made because I have never made a pie chart before. This is my one and only career pie chart, and I thought it was a nice way of illustrating. Now, it might seem to you like this piece of pie is a lot different from that piece of pie, but in fact, there is no statistical difference between the 2 studies. This estimate, the smaller estimate of 15%, is within the confidence interval of the 29% estimate. So the conclusion from these population attributable fraction studies is that the proportion of gay men who are gay because of older brothers is a minority, but not a trivial minority of gay men. And one last point to be made with regard to this particular statistic is that there is no point to be gained by trying to get ever more and more precise estimates of how many gay men are gay because of older brothers. And the reason there's no point to that is that that proportion is going to change as average family size changes in the population you're sampling from. a lot of my data came from baby boomers who had relatively large families. If we were talking, well, I guess it's easy to see how that would work in what we might call the degenerate case. Imagine if a one-child family were rigidly enforced, as China tried to do it. Well, there would be no gay men who existed who were gay because of older brothers, because there wouldn't be any older brothers. So you can easily see that average family size has to affect, not only has to affect how many gays are gay because of older brothers. Another way of talking about how important is this effect is to look at how widely has it been demonstrated. And it's been demonstrated in Western countries, in North America, in England, in Spain, Italy, and the Netherlands, and in one non-Caucasian population, which is the Samoan, independent Samoa, the population that Paul Vasey would have talked about when he was here. So he did actually 2 studies on the fraternal birth order effect. He added the results of the second one to the first one, but both studies showed a fraternal birth order effect in this Polynesian population, which is genetically quite remote from the Caucasian population. So this is one of the most broadly demonstrated phenomena in the field of sexual orientation. And the subjects were also born, I might point out, the subjects were born over a 130-year period. My earliest subjects were born around the time of the American Civil War. These would have been from databases I used at the Kinsey Institute, and these were guys who would have been old men at the time Kinsey started doing his interviews in the 1930s. So that's how I happen to have subjects that go back to, I don't know if they were gay or not, I don't remember, probably not. But at any rate, my subjects go from around 1860, I think one was actually 1859, up to roughly 1990. So it covers quite a broad historical period. And most of the samples were ordinary community volunteers, just plain vanilla gays and straights from the community. But we also have certain boutique subpopulations, such as transgendered patients, pedophiles, and hebephiles, who are men preferentially attracted to early pubescence, and sex offenders against adults. So the subjects have also varied in characteristics other than geography and chronology. This should now be hopefully halfway through the lecture, and I'm now going to turn from the basics of the empirical findings to the theory I've advanced to explain those findings. The theory is, I've called it the maternal immune hypothesis, which I regret in retrospect. I should have called it the maternal immunization hypothesis, but nobody stopped me, and now I'm sticking with it for the sake of consistency. And the idea is that this phenomenon reflects progressive maternal immune response to some male-specific substance, and an immune response on the part of the mother that's influencing the outcome of later pregnancies. Here's a graphical depiction of this hypothesis, which was made by a guy named Dave Putz, who actually was at that time a postdoctoral fellow in Mark Breedlove's lab. And I think Mark might actually be an author on the article in which this graphic was published. Here's how it works. And I'm going to use some deliberately vague language at this point, because then I have to fill you in on some immunology, which will be hateful because it's, but we have to go through it. And then I'll come back to this again and fill in what I'm going to deliberately use vague language for right now. Here we see a pregnant mother. I don't know how well you can see the colors here. Yeah, okay. We got a pregnant woman here, and she's got a male fetus. Now, I don't know whether or not you can see these little blue triangles. Can you see them? Okay. The little blue triangles are meant to represent substances that are present on the surfaces of male cells and are not present on the surfaces of female cells. And this stuff exists. So I'm not making this part up for my theory. We already know that there are such substances. Here we see that these male substances have gotten into the woman, perhaps at childbirth, when you have a lot of tearing of maternal tissue and a lot, relatively speaking, of fetal material gets into the maternal circulation during childbirth. So what's happened is that this blue stuff, which represents male material, has gotten into the mother, and she's developed an immune response to it, which are represented here by these little black Y-shaped objects. And I will explain to you later what these black Y-shaped objects are and why they're represented as Y-shaped objects. And some of these little Ys are touching the blue triangle with their very tip, which symbolizes that that Y-shaped object has grabbed some of this male invader stuff and is inactivating it, neutralizing it. Now, in the third slide, the third panel, the woman has now cleared out all that blue stuff. So it's all gone now, but she still has these things which are prepared to act against male-specific substances. And in the fourth panel, she's pregnant again. These Y-shaped agents of her immune system have crossed the placental barrier into the fetus's personal space, let's call it, and they're now looking for this male-specific stuff on the fetus, and where they find it, they're attaching to it and neutralizing it. Now we come to the painful part. Okay, I'm sure that everybody in this room has heard of antigens and has heard of antibodies, but probably could not tell you what those things really mean. An antigen is defined in a fairly circular way as a foreign substance that produces an immune response. An antibody is a blood protein that's produced in response to antigens. The other things, I won't even try to define them now. I'll show you in pictures. Here's a picture of an antigen. This whole thing is an antigen. Antigens are usually big molecules like proteins. So the thing as a whole is an antigen. On the surface of the antigen, we have these little knobs and gadgets, which are called epitopes. And these are the parts of the antigen that an immune system actually sees when it's recognizing that protein as a foreign invader. The immune system doesn't see the whole molecule. It sees these things on the surface, and that's what it's recognizing when it says, wait a minute, this isn't me. You know, this is a foreign thing. Okay, and I guess the only other point to be made is that epitopes are not actually orange, they're just drawn that way. Now, in many cases, an antigenic protein, or a protein that's antigenic to organism B, often forms part of the cell surface membrane in the organism it came from, organism A. And this is a depiction of how antigens look in the cell. They have a part that kind of sticks out of the cell and is waving around in the world outside the cell, and they have another portion that goes through the cell membrane and kind of dangles into the inside of the cell like a root. Now, why in Dave Putz's diagram were those things drawn as Ys? Well, because antibodies are basically kind of, sort of shaped like Ys. They are normally diagrammed as Ys when you look at, books or explanations of the biochemistry of antibodies. And this is a representation of kind of what they might look like if you could see them. It's sort of a Y-shaped thing. And this slide is the beginning of explaining how an antibody neutralizes or inactivates an antigen. The antibody has has something at the end of it which fits over an epitope on the antigen. So in this particular case, the antigen has an epitope. That's the visible part of the antigen, which is kind of shaped like a pyramid. And this antibody has a notch at the end of its arm that would fit over a pyramid. And that's how the antibody clamps onto an antigen, because something at the end of the antibody fits right over something on the antigen. That's how they connect. Different antigens can attach to different antibodies just according to what the shape of the epitope is on the antigen and the shape of what's at the arm of the antibody. Oh, I skipped a slide, damn it. Wait. Yeah. Okay. So here, for example, here we have an antigen with peg-shaped and knob-shaped epitopes. So this antigen could be bound by either this antibody, which would fit over the pegs, or by this antibody, which would fit over the knobs. Whereas this antibody here, which has kind of a V-shaped arm, could only inactivate that antigen, which has these pyramid-shaped epitopes. So that's how antigens and antibodies find each other, or how antibodies find antigens, because that's who goes looking for whom. And they connect by the shape of the epitope and the shape at the arm at the end of the antibody. This part will be over soon, so bear with me. We're going to zoom out one level now and look at a whole cell. This brown, big capsule-shaped object is meant to represent an entire cell. Here, these areas of darker brown represent cell surface antigens, and these little domes and blocks and pyramids represent the epitopes that are on the cell surface antigen. Up here, we have an antibody which has found one of the pyramid-shaped epitopes, and it's clamped on. And over here, we have an even luckier antibody which managed to grab a dome-shaped epitope with each of its arms. Okay, a little more background science, then we get back on the main track. It is known that there are some antigens which are specific to males, which either are different from their homologous form in females. In other words, the male can have a protein that has a corresponding protein in female, but they're not quite the same. And there are some male-specific antigens that don't even have a counterpart in females. And at least 27 proteins or families of proteins are known to be male-specific, and these proteins are encoded by genes on the Y chromosome. In many cases, the name of the protein and the name of the gene is the same, so I'll kind of be using these words interchangeably because in this field, the proteins and the genes are usually called by the same name. Of these genes, 3 are known to be expressed in fetal brain, and 2 of them encode cell surface proteins, whose names I've given there, PCDH11Y and NLGN4Y. Okay, so moreover, we know that, as I already told you, stuff gets from the fetus into the maternal circulation, and it's known that male cells have been found in a woman up to 27 years after she delivered the male fetus. So probably the great bulk of male-specific material gets cleared right away, but some of it can stick around for a while. And it's also known that maternal antibodies can and do cross the fetal compartment back into the fetus and can therefore get into the fetal brain. So I now want to revisit Dave Putz's diagram, but now using the real words instead of the vague terms I used before. Here we revisit the pregnant lady, and we're now going to call these blue triangles male-specific antigen. Here, the male-specific antigen has gotten into her system, probably at childbirth, because she's been torn in places, and she's developed antibodies, the Y-shaped black things, to these antigens, and some of them are inactivating the male-specific proteins because the end of their arm is meant to fit over an epitope on the antigen. Here is the immunized woman. I guess she's in between pregnancies now, but still has some detectable level of anti-male antibodies. And here she's pregnant again with a male. She's passing antibodies into the placental compartment and into the fetal compartment, and these antibodies are going looking for male-specific proteins, which they then inactivate by clamping onto them in the way that I demonstrated. Now, the maternal immune hypothesis would not be very convincing if I simply ended with this statement, and so anti-male antibodies get into the fetus and somehow make him gay. There has to be at least a plausible mechanism at the neuronal level that could explain how could an anti-male antibody affect a neuron in the fetal brain, and how could they affect it without killing the cells in the fetal brain. Well, let's start by asking, to build that part of the theory, you could start by asking, what's the purpose? Why do male fetuses have male-specific proteins that are preferentially expressed in brain in the first place? Are they there because at some earlier stage of evolution they were there and nature didn't need to clean up? Actually, that's unlikely because one of these proteins is a relatively recent development, and as far as I know, is only found in hominids. But it's conceivable that there are male-specific proteins expressed in brain or fetal brain, and they don't do anything. But it's also feasible that the purpose of these male-specific proteins that are preferentially expressed in fetal brain is that they are meant to be part of the brain organizing itself in a male-typical fashion. So we can say maybe that's why those proteins are there in the first place. Some biologists who do research on male-specific proteins have in fact speculated that that's why you have some of these proteins in fetal brain, although these speculations were kind of speculating at a general level that they're related to the development of male typical behavior, because immunologists do not get into sexual orientation. So they just talk at a general level about the development of sex dimorphic behavior. They don't get into sex dimorphic behavior, including the attraction of men for women. Okay, and we also know that some of the proteins, the male-specific proteins expressed in fetal brain, belong to families of proteins that are involved in recognition between cells and interaction between cells. Yeah. So all that has to happen for anti-male antibodies to influence the brain development is just bind to male-specific molecules and prevent them from playing their customary role in sex dimorphic development in the brain. They don't have to kill the male neuron. And in fact, the binding of antibodies to antigens often has no direct deleterious effect on the cell it's attached to. So the fact that, if people would say, well, if such an immune response were true, these fetuses would be dead, or else they would just be grossly impaired in some cognitive way. That isn't true. The simple fact of an antibody binding to certain male neurons does not mean that there would have to be signs of gross cognitive impairment or something more drastic like fetal loss. Now, this graphic, which is, again, made by somebody else, this graphic was made by a research immunologist named Chao Wang, who's currently doing postdoctoral work at Harvard. And she's an immunology consultant on the first laboratory study of the maternal immune hypothesis, which I'll talk about shortly. Now, what you see in the graphic here is, okay, on the bottom here, cell B, we have a cell which has male-specific proteins embedded in the cell surface. And she's indicated here that these proteins might be NLGN4Y or PCDH11Y, which are our two favorites. They're our favorites because they're preferentially expressed in brain or fetal brain, and they are cell surface proteins, which would mean they could easily have a role in cell-to-cell hookup, and also this makes them readily accessible to antibodies. So these are the ones we like, and these are the ones she's put in here as possibilities for these male-specific proteins that are hanging out of cell B. Up here we have another neuron which is meant to connect to neuron B, and it has proteins which are called binding partners, which are meant to connect to these male-specific proteins. Now, on the left, we see what would happen if there are no antibodies. This protein here would just fit into this this thing here. They're meant to connect in that way. But what happens if we have antibodies in the picture that are gumming up the male-specific protein? Well, when you start to have antibodies, again, our familiar Y-shaped entity, when you start to have antibodies that are glomming on to the male-specific protein, that protein can no longer connect with its binding partner in the other cell. So what this means is that the cells could not hook up, could not connect together in a male typical way, or in a way that they would connect without interference. These cells would just do whatever developmental thing they would do as if this cell surface protein were not there in the first place, which is to say, as if it were a female cell. So those are the basics of the theory. And of course, people want to know, has this been tested yet, or are there plans to test it? In fact, we are now doing the first attempt to do a lab test of this theory. And the test is very simple in principle. In principle, what we're doing is, and we've started doing this, we're recruiting mothers who have at least one known gay son and mothers, all of whose sons are heterosexual. We draw blood from these women, and we will eventually run assays and look for evidence of a maternal immune response to male-specific antigens. If the theory is correct, we should find more evidence of an immune response to male-specific proteins in the mothers of the gay men than in the mothers of the straight men. Now, the research is as difficult in practice as it is simple in theory. For one thing, we had to make educated guesses about which proteins to look at. I've already previewed which proteins are on our short list. Of course, you also then have to find a company that can make the proteins for you. We have one company which has been able to synthesize all of PCDH11Y, which is my personal favorite, and another company has been able to synthesize the extracellular portion of NLGN4Y, but not the intracellular portion. But the extracellular portion is the bigger part anyway. So I think that we're okay with that. So that's one aspect, is what protein are you going to look for? The other problem, however, is that if you have an immune response to anything, and it's an antibody response. There are other kinds of immune responses. But if you have an antibody response, that decreases over time. If you have a cold today, you might have antibodies that are detectable next week. They might be detectable in 6 months, but they might be undetectable in a year. And our study design, which looks for mothers of adult gay men, means these women might, in fact, have had an immune response that you could have seen if you got to them while they were pregnant or a year after, but it's now 20 years later, and those antibody levels might have declined to totally undetectable levels. So in order to get around the second problem, we're doing 2 different kinds of assays, and we're developing the assays as we recruit the mothers. The first type of assay, the ELISA, will look for current levels of circulating antibodies. And I'm not hopeful about this for the reason I just explained, that even if there was an antibody response, there not necessarily will be any antibodies detectable when the woman's gay pregnancy, so to speak, is now 30 years in the past. However, we're doing a second type of assay, which in theory can detect any immune response that a person ever had by re-stimulating memory T cells or memory B cells. And this kind of assay is called an ELI-SPOT assay. The second kind of assay is much more complicated and much more difficult, but we're going to try doing we're planning to do the second kind of assay as well. And it's complicated because you have to rush the blood from the mother to the lab. You only have a few-hour window because you've got to keep the cells in the blood viable, and then you got to centrifuge them out and put them in liquid nitrogen and all this stuff. So it's a hard thing. But we'll see what happens in a couple of years. And if it doesn't work, I'll say, well, it's because it was too hard. So that's it from my prepared remarks, and I can take any questions that people have.
So I'm sure people will have questions, and so let me ask you to please wait for the microphone so everyone can hear.
Thank you very much. It was a very interesting presentation. And you said at various points you followed the methodology of having a heterosexual group and a homosexual group. But for this part of testing the underpinnings of the theory, couldn't you go to the general population, look at women as they're delivering, shortly after delivering? Some will have had no previous boys, some will have had many, and to look at titers of antibodies, which will be, one would assume that they'd be in plenty supply around the time of delivery.
The kind of study you're talking about has been done with women who were like 8 years postpartum or 5 years postpartum, and these studies did in fact find higher titers. I'm not sure if they looked at antibodies. They might have used one of these other restimulation techniques for memory cells. I can't recall offhand. Essentially what you're saying can be done has been done, but these were mothers, like I say, who had like maybe 5 or 8 year old kids, so the kids hadn't declared their sexual orientation yet, so that part of the design couldn't be executed.
No, but at least to see if in fact as the number of male offspring increases, perhaps the titer level.
It has been shown comparing women who've had 0, 1, and 2 male fetuses. There has been, the immunological response has been shown in such mothers.
So you know that the mothers are showing an immune response to some?
Yes.
But not necessarily these two favorites?
Not preferentially to gay sons.
My question is this: so if a younger gay sibling had an older brother who passed away prior to his birth, then those antigens and stuff would still be carried on in the mother and have the same effect, perhaps. Okay.
Correct.
I have a second question. Have you studied populations of types of people, such as perhaps Mormons, strict Catholics, strict Christian groups, or else other countries who have much larger families and maybe have a stronger effect on this?
It's a good question. A population with larger families would, in theory, show a stronger effect. The closest we've come to that is the data collected in independent Samoa. In independent Samoa, mean family size is something like 6, which is humongous by Western standards. So the studies done in independent Samoa did actually have very nice results.
And I don't know if this is what you're getting at, but another possibility is that a previous pregnancy that did not result in a live birth, if it happened to have been a male, might have caused an immune response. And so from that regard, your estimates of number of gay men who are gay because of older brothers wouldn't detect those cases. You might not know those cases when the mother.
It probably would mean that I'm lowballing the estimate. What we did, you have to make a decision when you're asking people about their mother's reproductive history, because people don't know how many miscarriages their mother had and stuff like that. So we had to draw the line somewhere. So we would ask them to report numbers of siblings born alive. And then I just said, look, we can't know about miscarriages or early abortions, so we're just going to say, please report on siblings born alive to your mother that you know about.
Is there another question over here?
Yes, were any of your testing done with a control group?
The control group were heterosexuals. I'm not sure.
Let's see, but you're also looking at women who carried only daughters to look for an antigen response. So your expectation is they should not be showing an antibody.
Well, I think it's a matter of how far our money is going to go. For the critical groups are women who have given birth to at least one gay son and women who have given birth to only heterosexual sons, because we want to detect we're looking for homosexuality, not looking for differences that might be from anybody who's born a male, which we already know there would be some difference. So if we have the money, we'll throw in a group of, I don't know, middle-aged virgins or women who've only given birth to daughters. But we're doing this on a shoestring budget by American standards, so we'll see how many women we can get. Nuns would be good.
So I was just wondering if there are different reasons for why a man might be gay, namely, whether the genes as they are alone to make them gay without older brothers, or whether older brothers are necessary to cause that effect. Do you know if is there any reason to suspect that there might be some substantive nature or substantive difference in the nature of how homosexuality plays out in those two different groups?
Okay, the question is, are there differences between gays who might reasonably be put in the probably gay because of older brothers bin versus gays who are probably gay for some other reason? I only found one possible difference, and that is birth weight. I had a couple of different studies where I had data on birth weight, and it looked like gays who had multiple older brothers had lower birth weights than gays who had few or no older brothers. That's the only thing I ever detected. So that's conceivably, the birth weight stuff was consistent with the fact that the different roots to homosexuality might have kind of different associated features.
Let's see, I know there was a question here and then a couple there.
You mentioned that the mother gets the antibodies because of the exposure that usually happens during the tearing of the actual birth. Does that mean that not every woman would get those at all? I guess what I'm asking is, every time a woman has a son, is she going to produce those antibodies, or only if there's tearing during the pregnancy and there's more exposure?
Those are good questions, but that kind of exceeds what I have data to answer. Someone recently asked me a similar question to yours, which is that has someone looked at women who have cesarean sections, which might result in more entry of fetal material into her circulation? And the answer is no, I never even thought of looking at it, and I don't know the answer. They're good questions, but again, a lot of these data were based on archival data, so I was happy if it had sexual orientation and family structure, and these other kinds of refinements were just not in the data sets.
I'm the mother of a gay son, and he is dating a wonderful gay man who is the oldest of 4 brothers. So it's like the opposite scenario. His brothers are all heterosexual, and he's the oldest. What was going on with his mother's body? It's like the opposite.
Well, as I tried to stress, I think the fraternal birth order effect only accounts for in the neighborhood of maybe 15 to 30% of gay men. The other gay men, the cause is coming from something else: genes, fluctuating hormone levels during uterine development. So a gay man who has no older brothers or is an only child is from that great majority of gay men who can't be explained by the hypothesis I just outlined.
Thank you.
That's somebody else's job.
I just want to comment, when I'm talking about this, I need to point out to people that of course there are lots of oldest sons and only sons who are gay, but that's because there are a lot more first sons than any other kind, right? By definition.
I was wondering, I know that I study mainly animals, but I know there's colostrum produced by females after, soon after the pregnancy, that allows them to impart some of the antibodies that they produce to their offspring by through feeding methods. And I was just wondering if there's been any correlation between the amount of time a male spends breastfeeding and the chances of him being homosexual.
You know, to my knowledge, no one has ever looked at even whether a person breastfed and their sexual orientation, let alone how long they breastfed.
But I think we do know that there are antibodies in breast milk, right? And so that's a plausible mechanism.
So you mentioned that the antibodies tend to clear out over time, and most available right after delivery. So is there a spacing effect on the birth order effect? Does the number of older brothers effect decline as the spacing increases?
That's an excellent question, and I devoted one whole study looking just to the interval between births. I didn't find any effect whatsoever of the let's say I think maybe I looked at guys where the previous sibling was a male, okay, and then I looked at how many years were there between the birth of the previous sibling and the birth of the subject, and I didn't find any effect whatsoever. Now, this doesn't necessarily argue against the theory, because once you've been immunized to anything, your body will gear up to produce those antibodies much faster the second time around. So the interval between births it's not really, it would have been nice if there had been an interval, but the fact that there's no interval effect doesn't tell you much, because once you're immunized, even if your antibody level has gone back down to undetectable, you can regenerate those antibodies real fast with a second exposure.
Which is why immunizations work. And related to that, I mean, something else, right? I think so. Another question I think we had at one point was, once a mother has had a child that's gay, does that mean that the odds of the next son being gay are any different than if she had, i.e., is there some evidence that mothers are, becoming more likely to produce gay sons? And what do you know about that?
I guess the idea is that if the maternal immune hypothesis is correct, that once this thing is kicked in and a woman has had a fourth son who's gay, then now the thing has happened and all the further sons should be gay. I haven't been able to find evidence that it works in that predictable a way, which I once tried to address this by looking at what happens with twins, where one twin is affected by infections or something like that. And even twins aren't affected identically. So even when you have twins in the same uterus at the same time, one can be affected by some environmental event and the other one isn't. So I don't know that this would work in such a clock-like fashion that, bang, I've had one gay son and now all the rest are going to be gay, because things just don't work that way.
I'm a physician, and many years ago I used to deliver babies, and this is analogous to, it sounds analogous to Rh factor. And so when once a woman delivered a baby that was Rh negative, then we gave her for the next pregnancy Rogaine to prevent her from hurting that baby with the antibodies that she created as a result of that exposure. So have I got that right? Is this a similar kind of theory? And also, we've heard other speakers, particularly speakers from Europe in this series, talk about the concerns that some of the populations there have had about, like, what kind of manipulation could be done with this data. And I don't think it's ever seemed logical to me that we really could intervene in some way, but if this were true, there really could be an immunization of being against this way of becoming gay. Does that make any sense? And have you thought about that at all? And I'm not sure how.
I understood both your questions. With regard to the first one, yes, this is analogous to the birth order effect in hemolytic disease of the newborn caused by rhesus incompatibility, except that, of course, in that clinical scenario, it's much stronger than what I'm talking about because that's pretty mechanical. You know, like by the third pregnancy, the fetus dies without intervention. But the main difference is this only works, this is selective on male fetuses, whereas hemolytic disease of the newborn, I think it's more or less irrelevant to the sex, although about 3 studies have found that immunization to Rh is more readily initiated by a male fetus than by a male fetus for whatever reason. With regard to your second question, yes, I've been asked many times about applications of this in some negative way. For example, well, might people start selectively aborting male fetuses after they've already had 3 sons because they figure, well, now, you know. And the answer is that if you recall one of the graphs I put up where I show that the probability of homosexuality triples from 0 older brothers to 4 older brothers. Yes, that's true, but you're going from 2% to 6%. So if a couple said, we've already had 4 sons, and you're pregnant again with a male, and we don't really need one that might be gay because there'll be no grandchildren, what's the percentage in it, and decided to abort, 94% of the fetuses they would be aborting would still be pre-heterosexual. So you could not base family planning decisions, benign or malignant, on this phenomenon.
Let's see, I think the question here.
So I didn't quite follow the response to the question about the interval effect. So if the antigens enter the mother's body during carrying at birth, for example, then in the case of a second son, what's generating the regeneration of the antibodies that would affect development in utero if the mechanism is known?
Yeah, you put your finger on something that I just didn't go into in my talk, which is that the main point at which antibodies could enter the main point at which fetal material gets from the fetus into the mother is at childbirth. But fetal material also gets from the fetus into the maternal circulation earlier in pregnancy, in normal pregnancies, and even more so if there's a little defect in the placenta. So most of the immunization probably would happen at childbirth, but the mother is getting fetal material from much earlier on in pregnancies, even when it's a completely healthy placenta.
Are the antibodies detectable in the males also? Could you test for presence there?
Well, no, because they would be long gone. I mean, if you're testing an adult male, if there were any antibodies that had been in his brain, I don't even know how you go about doing that without killing the subject. But I think any antibodies would have been long cleared by the time the subject had reached adulthood.
And I don't know, but since there are fetal cells still found in mothers 20 years after, you do wonder whether there are any maternal cells that could be found in sons that would.
That's a really good question, and I don't know the answer to it. Not only are free-floating, so to speak, fetal cells from males found in mothers many years after pregnancy, but there are certain clinical conditions where fetal cells actually will colonize maternal organs and can stay there for a while and sometimes cause clinical conditions, which I can't reel off right now, but this has been reported.
Let's see this question here.
Returning to animals, are there any animal models that have been used where we're perhaps not looking at sexual orientation, but on sex dimorphic behaviors, where we can have multiple, pregnancies, large cohorts of, what's been done in rats or other ways?
There was one totally obscure study done like in the 80s where pregnant, how'd they do this? They immunized virgin rats with male-specific antigen and then got them impregnated, and compared with the control group, their offspring all didn't mate when presented with receptive females. That's probably the only positive study I could think of. It would be nice if there were animal models, but in so many ways, most animals are so different in their reproduction. A lot of them give birth to litters. Humans don't give birth to litters. One of my favorite protein, PCDH11Y, only occurs in hominid species. In fact, it might only occur in humans. So if that's the hot protein, there would be no animal model because other animals don't have it.
Again, when we talk about biological influences on behavior, certainly other mammalian species have taught us a lot that have contributed to our understanding of some human sex differences. So I just wonder that animal models give us the possibility of manipulating, of holding constant certain variables, because this is a dramatic effect. You know, as you point out, it really doesn't flow from anything else that we focused on. And one wonders, how do we go with certainty beyond correlations?
Well, a couple times graduate students or postdocs have approached me with expressing an interest in doing animal research, and I've certainly encouraged them to try it with rats. One guy, I think, tried it with rabbits, didn't get anywhere. And so of course I would love to see people doing animal research and trying to work up animal models on this, but you know, this is beyond my power to make happen.
And it wouldn't be easy to get funding for some, and so all animal research is incredibly expensive. I mean, you can't just do it in your spare time, and I would predict you'd have a hard time getting funding from any federal agency to pay for it.
Hi. How do we go from altered proteins in the brain to a change in sexual orientation?
Well, it wouldn't so much be a change in sexual orientation as a, how can I put it? It would be the default sexual orientation doesn't get shifted into the male position, okay? The toggle switch doesn't get flipped over from the brain developing in a way that includes erotic attraction to males to the position where the fetus now has erotic attraction to females. And how that part of the theory was the slide where I showed how antibodies could prevent the connection of neurons that involve male-specific proteins.
Okay, I guess my question is then, what's the relationship between the proteins that you showed us and sexual orientation? What makes you think that these in particular would be significant in their effects?
You have to have, in first place, in my view, the only thing that fits the epidemiological data, the only thing that fits the pattern of older brothers, and specifically biological older brothers, almost has to be some kind of immune response, because the mother's body is clearly remembering the number of male fetuses she's carried and ignoring the number of female fetuses. Now, the only organ system with that kind of memory is the immune system, aside from the brain. So I think it's pretty clear that it has to be something about the mother's immune system. It's got to be an antibody response, or it doesn't have to be, but if it were the other kind of immune responses, like cell-mediated, probably the whole conceptus would be ejected. There'd be no child at all. And as far as why did I say these proteins? Because they're preferentially expressed in brain or fetal brain. In other words, there are some male-specific proteins that are primarily expressed in the testes. Why are they there? Because they're contributing to spermatogenesis. So why are these cell surface proteins in the brain? Probably to do something that's done by the brain and not by the testes. And I like the two ones I picked because they're on the cell surface where antibodies could get to them, and because they're classes of proteins that are known to normally have a role in the hookup of cells during brain development.
And of course, as to how any protein function necessarily contributes to a particular behavior is something. I mean, we know a lot about neuroscience, and we don't have any examples of that where we can tell you this protein, works in these cells and causes this change in behavior. That sounds so easy, and in fact, that's so difficult.
Just
just along a similar line there, then, has anybody looked at correlating gene expression of, or the expression of the proteins? You know, are there more proteins in certain men of your particular two proteins that you talked about, or is it more of the time? I mean, there could be lots of other factors, but are certain men producing more of this protein as a cell surface protein than others?
Things like that have been looked at with some male-specific proteins, but not the ones that I showed here. One of them that's very common, the first one ever identified, for example, has different names, but one of them is SMCY. That I found seems to be preferentially expressed, or more expressed rather, in older men compared to younger men for whatever reason. So these kinds of distinctions have been looked at in other male-specific proteins, but not the two that I emphasized here.
And I'm sure there are going to be more questions, but I think we should stop now. And so thank Ray again, and then come up and